Okay, so moving on to section 10 of chapter 19, we're going to talk about the blood flow through our capillaries and how it's slow and the purpose of the slow blood flow and its function in helping promote diffusion of nutrients, gases, and other fluids needed. So the velocity flow changes as it travels through our systemic circulation. We talked about this earlier as it leaves our aorta, goes to our arterioles, capillary bed, and then our veins. It's going to be fastest in the aorta and slowest in the capillaries, and then is going to increase again in the veins. Why? So speed is going to be inversely proportional to the total cross-sectional area. Well, if we added up the total area of all the capillaries making up our capillary bed, we would see that it has the largest total cross-sectional area. So because they have the largest total cross-sectional area, they are going to actually have the slowest flow because speed or velocity is inversely related to that area itself. Slow capillary flow is really important because it's going to allow adequate time for exchange between the blood and the tissues. It allows for enough time for the oxygen to be dropped off to the tissues and for the waste products to be picked up. We have something called vasomotion. This is just intermittent flow of the blood through the capillaries, and it's intermittent because we have the opening and closing of the precapillary sphincters. Okay, so a lot of molecules pass by diffusion, which is a type of passive transport between the blood and the interstitial fluid. If we're talking about diffusion, which is a type of passive transport, that means the molecules are moving down their concentration gradients from an area of high concentration to an area of low concentration. We have 4 different routes that these molecules can move across the capillary. So the first option is that they diffuse directly through the endothelial membrane. So things that can do this would be lipid-soluble molecules, such as respiratory gases. They can pass through clefts. So these would be things like water-soluble solutes, so things like sugars and amino acids. They can also pass through fenestrations, and these are the same type of solutes that can cross or pass through the clefts. So water-soluble solutes as well, so sugars and amino acids. Or the fourth option is that they can be actively transported by a vesicle. And if we're talking about fluid, that would be a pinocytotic vesicle or caveolae. So larger molecules, things like proteins, would have to be actively transported across the wall. So active transport compared to passive transport, if you remember, passive means that it doesn't require an energy source. It just uses the concentration gradient in order for things to move. If we're talking about active transport, we need our body's energy currency in order for things to cross. So something like ATP. So here's just showing you the 4 possible routes if we're talking about things diffusing across the capillary wall. One is directly diffusing across the capillary wall. So things that are lipid soluble can cross plasma membranes as long as they're pretty small. The other 2 that allow for water-soluble substances like amino acids are them moving through intracellular clefts, shown here, or the fenestrations that we talked about in the different types of capillaries. And then our final way is actually using a vesicle in order for things to cross. However, if we need to use a vesicle for things to cross, that's going to require energy because it's a type of active transport. So bulk flow. So fluid is forced out of clefts of capillaries at the arterial end, and most returns to the blood at the venous end. This is extremely important in determining the relative fluid volume in the blood as well as in the interstitial space. So if we're talking about bulk flow, we're talking about the fluid flow across the capillary walls, which causes continuous mixing of fluid between both the plasma and the interstitial fluid. So this helps maintain the interstitial environment. The direction and the amount of fluid flow is going to depend on two opposing factors. So we have hydrostatic pressures and colloid osmotic pressures. So hydrostatic pressure. Hydrostatic pressure is the force that is exerted by a fluid pressing against a wall. So we have two different types. We have capillary hydrostatic pressure, which is represented by a capital H and a capital P with a subscript of C to represent capillary. And then we have interstitial fluid hydrostatic pressure. Again, we still have our capital H, capital P, but the subscript is now IF to represent the interstitial fluid hydrostatic pressure. So our capillary hydrostatic pressure is the pressure that forces fluids through capillary walls. And it's going to leave behind cells and most of the proteins because they're not allowed to cross. If we're talking about interstitial fluid hydrostatic pressure, that's the pressure of the fluid pushing back into the vessel, usually assumed to be 0 because the lymphatic vessels drain the interstitial fluid. So if we're talking about hydrostatic pressures, the only real important ones are capillary hydrostatic pressure or that of the fluid within the capillary itself that's helping push the fluid through the capillary wall into the interstitial space. It's much, much lower if we're talking about interstitial fluid hydrostatic pressure because, again, it's constantly being drained by our lymphatic system. Our colloid osmotic pressure, also called onconic pressure, this is the sucking pressure created by non-diffusible plasma proteins, primarily albumin, pulling water back into the capillary bed. If you remember osmosis, water moves to the area of higher concentration. Because we are leaving the larger proteins behind as we're pushing the fluid out of our capillary into the interstitial space, we also have an opposite pressure that's being created as a result of leaving those proteins behind. And that is the osmotic pressure because water naturally wants to flow back towards the high solute concentration, in this case protein concentration that's left. So it's a sucking pressure created because those plasma proteins remain in the capillary themselves and they are wanting to pull water back into it. We also have the interstitial fluid colloid osmotic pressure, and this pressure is inconsequential because the interstitial fluid has a very low protein count. So we have to account for the proteins that are present in the interstitial fluid as well as the capillary itself. However, there's not a whole lot of proteins in our interstitial fluid, so we don't have a lot of pull of water from proteins that are in the interstitial fluid back out of the capillary. It's predominantly going to be an osmotic pressure for a pull into the capillary because that's where the high concentration of proteins are going to be. So in order to calculate our net filtration pressure, we have to take into account all 4 of these forces that are acting on the capillary bed. So our net filtration pressure, NFP, is basically the force pulling fluid out minus the force pulling things in. And if you can imagine, our goal is to push fluid out. Our pull out forces, which are these two, have to be greater than our pull in forces, which are these two, in order to continually push fluid out. So our 2 outward forces are our hydrostatic pressure of our capillaries and the onconic pressure of our interstitial fluid, which is not very high. So predominant outward force is going to be the hydrostatic pressure of our capillaries. So the fluid pushing against the capillary wall, wanting to go out into the interstitial space. Our 2 pulling inward forces are our hydrostatic pressure of our interstitial fluid trying to push fluid back in, not very high. But the greatest pull in the force is actually going to come from the proteins that are left behind in our capillary, so the oncotic pressure. So net fluid flow at the arterial end we refer to as filtration. The net fluid flow at the venous end is what we refer to as reabsorption. So more fluid leaves at the arterial end than we actually return at the venous end. And where did the difference go? It went to our lymphatic system. So if you need a review of hydrostatic pressure or oncotic pressure or osmosis and how things work, I recommend going in and reading this table and kind of understand what's happening. This is the same thing that we had in words previously, just showing you the different pressures. If this is the inside of our capillary here in red, you can see the pressures that are trying to push the fluid out. So the hydrostatic pressure in the capillary, or the pressure of the fluid itself pushing against the wall, is an outward force. Opposing it is the osmotic pressure in the capillary, so the pull of fluid back into the capillary because of the high concentration of proteins. Down here we do have the two other pressures, but they're not considered to be heavily influential on the net filtration pressure because they're very, very small. However, I would still recognize that these are pressures acting to drive fluid across the capillary. So this is net, kind of showing you. Again, you don't have to memorize specific numbers or pressures. Really just have an understanding of what our greatest outward pressure is, what our greatest inward pressure is, what net filtration pressure represents. So if we're talking about fluids leaving our capillary, that can lead to something called edema. And edema is basically swelling as a result of having too much interstitial fluid. So it can be caused by either an increase in our outward pressure, driving more fluid out of the capillaries and into our interstitial space, or a decrease in our inward pressure to help put some of that fluid back into our capillaries. So an increase in the capillary hydrostatic pressure accelerates fluid loss from the blood, pushing more of it out into our interstitial space. And this could be a result from our venous valves not working correctly, if we have some type of blockage in a local blood vessel, if we have congestive heart failure, or if we have too much blood volume. So an increase in interstitial fluid osmotic pressure can result from an inflammatory response. So inflammation increases the permeability of our capillary wall. If we increase the permeability, we're allowing more things to cross, like proteins. So then we can allow proteins to leak into our interstitial fluid, which causes a large amount of fluid to be pulled into the interstitial space, because a lot of times water follows concentration of proteins. A decrease in the capillary colloid osmotic pressure is going to hinder fluid return to the blood. Things that can cause this is hypoproteinemia. So if we have low levels of plasma proteins to begin with, that could also decrease that pressure. We would have low levels of plasma proteins if we have liver disease, if we're malnourished, or if we have glomerular nephritis, which means we have the loss of plasma proteins from our kidneys. So edema also can be caused by decreased drainage of the interstitial fluid. Typically, our fluid in our interstitial space is drained by our lymphatic system. So if for some reason we have a blockage or a disease, that can reduce the amount of drainage from our lymphatic system and lead to swelling in our interstitial space. Typically, you just go in and surgically remove whatever the blockage is. Excess interstitial fluid can also accumulate in our subcutaneous tissues, generally causes pitting edema. I believe I have a picture here shortly. Oh no, I don't. So pitting edema basically is if you have some sort of swelling and then say you push down on the swollen area, if it leaves an indentation from the pressure of your thumb, that would be referred to as pitting edema. And edema can impair tissue function as a result of increased distance for diffusion, because now we have more fluid in that area, so it takes a longer time for things to be able to fuse across a larger fluid space. Slow fluid loss can be compensated by our renal mechanisms or kidney, but if we have some sort of rapid onset, that can have serious effects on our circulation.